Create a fast-paced video for YouTube about phage and AI just crossed a line in biology, and Reddit is freaking out. Imagine telling an AI, design me a new virus. Not a picture of a virus, not a computer simulation, an actual genetic blueprint. Then imagine scientists take that blueprint, chemically build it, put it into the real world, and it works. That sounds like science fiction. But according to research from Stanford and the Arc Institute, that is essentially what just happened, with one extremely important qualification. These aren't viruses designed to infect humans, they're bacteriophages, viruses that infect bacteria. And the bacteria they targeted? E. coli. But here's where this story gets much bigger than AI kills bacteria. Because for the first time, we're seeing a complete loop. AI designs DNA. Humans synthesize the DNA, the biological system works, then scientists test what the AI created, and that changes the conversation around AI and biology completely, because we've spent the last few years watching AI generate text, images, videos, and computer code. Now we're watching it generate something much more consequential, biological instructions, and Reddit immediately picked up on the part that Stanford's headline doesn't fully capture. The question isn't just, can AI help us fight antibiotic-resistant bacteria? The question is, what happens when AI becomes a general-purpose design engine for biology? Let's break this down. First, what exactly did Stanford build? The system is called EVO2. It was developed by researchers including Stanford's Brian High, and it's part of a new class of biological foundation models. If you've used a large language model, you already understand the basic intuition. An LLM learns patterns in language. Give it, once upon a, and it predicts what might come next. AVO works with biological sequences instead. Instead of predicting words, it learns patterns in DNA, so you can think of it as something like a language model for the language of life. But there's an enormous difference between generating a paragraph and generating a genome. A paragraph can be wrong and nobody dies. A biological sequence has to obey the rules of biology. Genes have to work, proteins have to function, genetic elements have to interact correctly. The entire system has to be viable. And that's why this experiment is so interesting. The researchers didn't simply ask EVO2 to modify one gene. They challenged it to generate whole bacteriophage genomes, and they picked a very interesting target. The starting point was a bacteriophage called phage 174. Don't worry about pronouncing that. Just remember, it's a tiny virus that infects E. coli. Its genome is only around 5,400 DNA bases long. That's microscopic compared with something like the human genome, which contains roughly 3 billion DNA base pairs. So the researchers weren't starting with something remotely as complicated as a human genome. And that's important, because this was a proof of concept. The question wasn't, can AI design a human organism? The question was, can a genome model generate an entirely new, functional viral genome? And the researchers had a very clever way to find out. AI generated the designs. Then biology got the final vote. The researchers generated thousands of candidate genomes computationally. They then filtered those candidates using biological criteria. The most promising candidates were chemically synthesized, and then they were tested against E. coli. This is the part that makes the story different from most AI biology headlines, because the AI doesn't get to grade its own homework, the laboratory does. Out of hundreds of synthesized candidates, 16 AI-designed phages were found to be viable and effective in the experiments. And some showed higher fitness than the original natural phage they were based on. That's a big deal, because it means the model wasn't simply copying a known biological sequence, it found designs that could actually function. The researchers described substantial evolutionary novelty in these genomes, and one of the fascinating findings was that some designs contain changes that aren't found in known natural sequences. So this isn't simply, AI found an existing virus in a database. It's closer to, AI explored biological possibilities that evolution hadn't obviously handed us in an existing sequence. And that is where things get interesting. But here's the first myth we need to kill. You may see headlines saying, AI created viruses from scratch. That's catchy, but it's not the most scientifically accurate way to describe what happened. The researchers weren't asking EVO to invent biology from a blank screen. They used a known phage as the design template, biological constraints, and information learned from genomic data. So this isn't, AI spontaneously invents a virus from nothing. It's, AI learns biological patterns and generates novel sequences within a biological design space. That distinction matters, because the achievement is still enormous without the hype. The real breakthrough is that the generated genome crossed the line from computer output to functional biological system. Now let's talk about the E. coli part. Why would anyone want AI-designed bacteriophages? Because bacteria are getting harder to kill. Antibiotics have transformed medicine, but bacteria evolve. A drug kills susceptible bacteria, the resistant ones survive, they reproduce, and eventually the treatment becomes less effective. Phages have a different strategy. They're biological predators of bacteria, but phages have a problem too. Bacteria can evolve resistance to them. So imagine you have one phage. The bacteria figure out how to escape it. Game over. But what if you have multiple genetically distinct phages attacking the bacteria through different mechanisms? Now the bacteria have a much harder problem. That's the idea behind a phage cocktail. And Stanford's researchers demonstrated a proof of concept using their AI-designed phages. A cocktail of 16 phages was able to rapidly overcome resistance in E. coli that was resistant to the natural FX174 phage. And this is where AI could become extremely useful, because instead of spending years trying to discover one perfect phage, you could potentially generate and screen huge numbers of candidates. AI proposes, the lab tests, the successful designs go forward, and the failures get discarded. It's the same basic philosophy that's transforming drug discovery. Search a gigantic design space computationally, then test the most promising candidates experimentally. But Reddit noticed something important. If you look at the recent discussions around this research, the reaction isn't simply, wow, amazing medical breakthrough. A lot of people immediately went, wait, AI can now design functioning viruses? And honestly, that's a reasonable question. One of the newest Reddit discussions about the research had thousands of upvotes, and the comments quickly moved from medical applications to biosecurity. Another discussion basically framed the technology as a major milestone precisely because scientists can now generate viral genomes and test them in the real world. And that's where the story gets uncomfortable, because the same capability that could help us fight bacterial infections could eventually be used in ways we don't want. The researchers themselves acknowledge this dual-use concern, and the paper explicitly discusses the possibility that future-generated genomes could encode harmful biological functions. So this isn't some criticism Reddit invented, the scientists are worried about it too. But let's not overreact either. There's an important distinction between AI designed a bacteriophage that attacks E. coli and AI can now design a human pandemic virus. Those are absolutely not the same thing. The demonstrated organisms in this study are bacteriophages. They target bacteria. They are not human-infecting viruses, and EVO2 was designed with biological safety considerations, including exclusions around eukaryotic viruses in its training approach. So no, this experiment does not demonstrate that someone can open an AI chatbot tonight and generate the next pandemic. That's not what happened. There is still a massive gap between generating a sequence and successfully creating a dangerous pathogen, but here's the uncomfortable part. That gap is exactly what researchers are going to be watching. Because technology doesn't stay frozen. The models get better, the genomes get longer, the biological systems get more complicated, and DNA synthesis becomes increasingly accessible. That's why the safety discussion needs to happen now, not after the capability becomes routine. And there's another misconception. Some headlines make it sound like AI just solved antibiotic resistance. No, absolutely not. Bacteria evolve, phages evolve. Resistance is an evolutionary arms race. The Stanford experiment demonstrates a promising strategy for overcoming resistance in a specific experimental setting. It doesn't mean we're done with superbugs, and phage therapy has another challenge: specificity. An antibiotic may target a broad class of bacteria. A phage can be much more selective. That's sometimes incredibly useful. But it also means the future of phage medicine could look very different from taking a conventional antibiotic pill. Imagine a future workflow like this. A patient has a serious bacterial infection. The pathogen is identified. Its resistance profile is analyzed. Researchers select or generate compatible phages. Multiple phages may be combined. The treatment is monitored. And if the bacteria evolve resistance, you potentially redesign the cocktail. That's much closer to programmable antimicrobial therapy than traditional antibiotics. And AI could make the design side dramatically faster. Here's the part that really blows my mind. Think about how drug discovery traditionally works. Nature gives us molecules. Scientists discover them. They modify them. They test them. But nature only gives us the biological possibilities that evolution happened to explore. AI doesn't have to limit itself to those exact sequences. It can learn patterns across enormous biological datasets and generate new combinations. That's the same fundamental idea behind generative AI everywhere else. We went from AI recognizes patterns to AI predicts patterns to AI generates new patterns. Now biology is entering the same cycle. And suddenly the question becomes, how much of biology is actually designable? Could AI eventually design better enzymes, new metabolic pathways, microbes that manufacture useful chemicals, cells that perform specialized functions, new therapeutic proteins, and eventually, much more complex biological systems? That's where this becomes bigger than phage therapy. And this is where I think the Reddit discussion gets something right. The most interesting reaction isn't the people saying, "We're all going to die." That's too simplistic. The more interesting reaction is, the technology is moving faster than the governance, because here's the fundamental problem. Biology doesn't have a software undo button. If an AI generates bad code, you delete the file. If an AI generates a biological sequence, the consequences depend on what humans do with it. That's why the pipeline matters. Model, design, synthesis, experiment, validation. Every one of those stages becomes a potential safety checkpoint. And that's actually one reason AI could eventually help with biosecurity too. If AI can design biological systems, perhaps AI can also help detect dangerous designs, screen DNA sequences, identify suspicious biological functions, flag risky combinations, model potential biological behavior. In other words, the same technology that increases biological design capability could potentially become part of the defense system, but that requires us to build the safety layer deliberately. It doesn't happen automatically. So where does Evo 2 actually leave us? We're not at AI can create anything alive. We're not at AI has solved infectious disease, and we're definitely not at AI can casually create a human pandemic. What we have is something more specific, and arguably more important. Scientists have demonstrated that a genome language model can generate whole viral genomes that are substantially novel and experimentally functional. That's a real milestone, and it changes the role of AI in biology. Previously, AI was primarily helping scientists understand biological systems. Now it's beginning to help them design them. And once you can design something, you can start engineering it. That's the transition. The bigger race has already started, and here's where I think this story goes next. Today's experiment uses a tiny bacteriophage. Tomorrow's models will work with larger and more complicated genomes. Researchers will improve controllability. They'll try to specify traits. They'll generate more candidates. Laboratory automation will accelerate testing. And eventually, you're going to have something that looks less like AI-assisted biology and more like an automated biological engineering loop. AI generates, robots build, experiments test, results go back into the model generates again, and the cycle repeats. That's essentially an evolutionary search process, except now humans are building the search engine. And that could be one of the most powerful technologies of the next decade. It could help us fight resistant infections, it could produce medicines, it could manufacture chemicals and materials, it could help us understand biology in ways we've never been able to before, but it also creates a new category of risk. Because if biology becomes programmable, someone eventually has to decide what should and shouldn't be programmable. So here's my take. Don't look at this story and conclude, AI created killer viruses. That's sensationalized. And don't look at it and conclude, it's just another antibiotic breakthrough. That's underselling it. The real story is somewhere in between. AI has demonstrated that it can generate novel biological instructions that, when physically synthesized, can produce functional viruses that attack bacteria. That's the breakthrough. And the next question isn't whether AI can design biology. We've just seen evidence that it can. The next question is, how far can it go? And perhaps even more importantly, can our safety systems keep up with it? Because we've spent decades teaching machines to manipulate information. We're now teaching them to manipulate the instructions of life itself. And that is a very different kind of AI revolution. If you want to understand where AI is actually going, not just the latest chatbot update, subscribe. Because the next frontier isn't just artificial intelligence, it's artificial biology.